please join me in welcoming to the Distinctive Voices podium, Dr. Larry Cahill.
Okay, thank you very much. Thank you, I appreciate it. Thank you all for coming here, for taking the time to be here tonight. I plan to try and give you your money's worth. I might even do better than that. The topic tonight is sex influences on brain and emotional memory. 10 years ago, I've been studying emotional memory, how brains make emotional memories for about 30 years. About 10 years ago, I discovered that it actually matters if you're a man or a woman. Before that, I couldn't have cared less. I assumed it didn't matter. It's actually a very serious topic, but now it's moved center stage, and that's the story I'm going to tell you tonight. So this is a talk that's going to have 3 basic parts. By the way, just out of curiosity, will you please raise your hand if you've heard me lecture before? Okay, whew, not too bad. For a lot of you, for a few of you, it'll be a rehearsal. In any event, this is a lecture that's going to have 3 parts. The first part, I'm going to give you some background. How we think it is that brains make memories for emotional events. A very well worked out view, heavily anchored in animal research, but also supported by human subject research. And just as my research in that segued into studies of sex differences, so this talk is going to segue into part 2, into studies of sex influences on the brain. And I'm going to tell you a little bit about why this topic is so hugely important for neuroscience, even though a lot of neuroscientists don't realize that yet. But it's coming like a tidal. It's a tidal wave. It's a tidal wave that's coming. And then finally, having done that, I'm going to take you to the third part of the talk, take you back to my lab, and tell you about sex influences we've been finding on brain and memory now that, as I put it, the blinders have fallen off. And we've stopped assuming that sex doesn't matter, and we've started assuming that it darn well might and probably does. So let's start with the background, emotional memory. We all intuitively understand and have experienced that the things in our lives that tend to have a little bit of an emotional punch tend to be better remembered. Not just unhappy things, but happy things, basically things that tend to be a bit arousing. They tend to be better remembered. Not perfectly, but better remembered. And here's a specific example. I took this from the Los Angeles Times about 10 years ago. This is a woman named Holly Viola. She witnessed a suicide jump in Seattle in 2001, and she had this to say. She said, "We were shocked. This person was going to dive before our eyes. Our hearts were thumping for probably hours afterwards." I don't know Holly Viola. I clipped this from the newspaper 10 years ago, but I'm willing to bet you that Holly Viola remembers this today, right? Okay, I don't think there's any argument. Now, does she remember it perfectly? Absolutely. No, there might be some false parts in there, but she remembers it. You think she remembers much from what happened 3 days before or 3 days after? Unlike the hyperthymesia patients, who I'm not going to be talking about, who can remember most every day of their life, they're a phenomenon, Holly's like the rest of us. She doesn't remember much of her life, but boy, she tends to remember that which had a bit of an emotional punch. Her heart was pumping for hours afterwards. And in fact, this heart pumping business is a critical part of how we think it is that your brain is making memories for emotional events. Heart pumping and acting through a little structure known as the amygdala. See that little guy shaped kind of like an almond? It's inside, a couple inches in from your ears on either side of your brain. You have 2 of them. And it's called the amygdala, Latin for almond. Looks like a little bit like an almond. Looks kind of more like a grape in this picture, but imagine an almond, okay? But take a look at the wiring of this almond. Over here, you see a wiring diagram of the primate brain. What's connected to what? And if you just look at that picture, you can see in the middle is the amygdala. It is amazingly widely connected to everywhere. It is beautifully suited to tell everybody else, hey, crank it up, remember that better. It's beautifully suited, positioned to modulate. And that's exactly what we think it does. We think that this little amygdala, working in concert with this thumping stress hormone milieu, acts during and after an emotionally arousing event to tell other places in your brain, like your hippocampus or your cortex, hey, that was more important, crank it up, store that better. And that's the fundamental idea that I've been working with now for about 25 years on how it is that the core of how it is that we think our brains naturally, effortlessly take emotional arousal and turn it typically into strong memories. And it's the idea, the brainchild most heavily of my colleague Jim McGaw here at Irvine, who many of you certainly know. That's Jim on the left there, shown interestingly with Alan Alda, who came about 6 or 7 years ago to film a PBS special on our research, which was quite nice. And it's an idea that is heavily anchored in animal research. So, for example, this has been a workhorse task that we've used to study hormones and memory. Little rat's placed in this side of a box. Little rat crosses over to this side, and when he does, zzz, little half-second foot shock. Rat doesn't like it. If you take the rat out, put him back in the next day, what do you think is going to happen? He's going to stay out, depending on how well he remembers, right? Well, if you take a typical rat with a typical foot shock and test them the next day, they look about like that. They remember. That's pretty good memory. But look what happens if you take a rat, give him the training, and then right after, take him out of the box and give him a shot of adrenaline. Look how well they remember the next day. Right? So that's like a classic study from 1975. There's been many, many studies since that have shown that your body's naturally occurring stress hormones, when released in an emotional situation, can feed back to your brain and boost memory storage. And in fact, a bit of a connection to this lecture, we've shown this in a number of different tasks, including a task that involves just a big tub of water, about a 6-foot wide tub of water. And we can enhance memory with adrenaline injections in that task as well. And I had the great pleasure one day, about 15 years ago, of showing that task to Arnold Beckman. And I'm just mentioning that because of the setting and everything here, but he was in a wheelchair at the time, and it was so charming to me because when I went to put that, he couldn't see what was going on because he was 2 foot down low. Well, when I went to put that rat in the water, so rat had to find a little platform. That's the problem they had to solve. Dr. Beckman, they told him to stand him up, and he hung onto the edge of the tank of water, and he wanted to peek over and watch what that little rat was doing. So just a little point of connection between my research and this lecture tonight. I was able to show, actually, Dr. Arnold Beckman some of these very tasks that we use. Well, about 20 years ago now, time flies, I returned from a postdoc in Germany to Irvine because I wanted to extend this work on emotion, stress hormones, the amygdala, and memory to humans, to the human animal. I basically wanted to know, is this true in humans or not? So I returned, I started working with Jim McGaw again to work on this topic. And so we did some things that you would probably do yourself if this is what you're going to do, some pretty obvious things. For example, is it really the case that adrenaline can enhance human memory, like it does in the rats? Well, if it is, then I ought to be able to teach you something, and just like the rat studies, inject you right afterwards with a shot of adrenaline, and if I get my conditions right, enhance your memory. And I was able to do that with my colleague, Mike Alkire, who's in the Department of Anesthesiology here at UCI. What we did was we had people watch a series of slides. They just watched the slides. Then right after, Mike injected them with adrenaline. Then we waited a week, and then we tested their memory. And it's a kind of a boring graph, but right here you can see some of the slides that turned out to be a little more arousing to begin with, we significantly enhanced memory for them. This graph right here is the world's first and still only demonstration that a naturally occurring human stress hormone can act to enhance human memory. Now, it turned out that injecting people with adrenaline is stressful to me. I have to have a crash cart in the lab. It's a mess. And so I decided I got to come up with I'm dying here. I got to come up with a better way, a more efficient way to turn on this stress response so I can study what it does to memory. And this is what I came up with. The cold pressure stress used by cardiologists for at least 70 years or more to see how the old ticker is functioning, all right? And we've now done a lot of studies in which we did much the same thing as the adrenaline thing, only with cold pressure stress. We have people watch some stuff, and right after we have them drop their arm in ice water, and we wait a week or so, and we see what they remember. I had the good fortune also of doing this to Leslie Stahl. I got a big kick out of that. I haven't been able to determine whether or not she remembers this. I'll have to email her producer. But I'm guessing she remembers it from the look on her face. So, anyways, that's what we did. Had people watch some slides, and then they stuck their arm in ice water or as a control, warm water. And we waited a week, tested them. And if you look over here, these are the people who stuck their arm in ice water. These are the people who stuck their arm in warm water. This is for the slides that were a little arousing to begin with. I'm going to sidestep that for you because it's kind of a side point. But again, we found, consistent with the rat research, that a little bit of arousal right after you learn something helped stamp the stuff in, helped make it better for memory. Now, what about the flip side? What about blocking the arousal response? Here we are turning it on with shots of adrenaline or sticking your arm in ice water, but what if we do the converse? Or is it the reverse? Converse, I think. What if we show something that's pretty arousing, but block the stress hormone response with a drug? So we did that. It was one of the first studies I did actually when I returned from Germany. We had people come into the lab and watch an emotionally arousing story, and the emotional bits happened in what we call phase 2 of the story. So it was a short story, a boy's walking with his mother, and in the middle, he gets hit by a car, and his feet are cut off and sewn back on. And that tends to be more memorable than either the beginning or the end parts. Now, that was not intended to be funny. Sometimes I can't help myself, but it's not funny when you watch it. In any event, when you and I come back a week later and I give you a surprise memory test for that 3-phase story I showed you a week ago, you and I tend to remember the emotional bits of the emotional story better. So this was my college sophomores who watched the story while on a placebo. And these are my college sophomores who watched the story while on a beta blocker. You've probably heard of beta blockers, a beta-adrenergic blocking drug used to suppress the body's adrenaline response. So this was very exciting. This showed us that the flip side of the story was also holding true in humans. That is, if something's emotionally arousing, but we block the hormone response, the stress hormone response, we can impair memory. So that was very exciting. Now, I'm going to come back to this later in the talk, but at the time, this was 1994, I published this. Remember, I told you I've been into sex differences for about 10 years. I just published, this was men and women together. I just published them together because who cares, right? Doesn't matter. Turns out there's a sex difference hiding in here, and I'm going to come back to that. Okay, well, let's finish up the first part of my talk, the background part. Besides the stress hormones, the other key player in our model of making strong memories is the amygdala. Come on, class. The little almond, right, in your brain. Well, what about that little almond? Is this also important for making strong memories for emotional events? And we've been able to study that in a series of ways. One way was I was able to find a fellow in Germany who had a rare disease called Urbach-Wiete disease that resulted in his brain being entirely fine, except he didn't have an amygdala. It's true. His brain is basically entirely fine, but his 2 amygdalae calcified, rotted away. Not good for him, good for me. I'm being facetious, but as a person who's interested in the function of the amygdala, right, that is about the perfect person you want to find. If you want to find, so it was worth flying to Germany, translating my little 3-phase story into German, and showing it to him. And here's what happened. The German controls, like you saw before, they remember the emotional bits better. The patient, he remembered the non-emotional beginning of the story just fine, but he just tapered off. He didn't show any boost in memory for the emotional bits like you and I do. We were able to find the same thing again on a second patient in Iowa. Well, that's all well and good to study people with brain damage, but as all of you know, there has been a true revolution the last 20, 30 years in neuroscience, brain science, and it's called human brain imaging. When I was a graduate student, the stuff I'm about to tell you, and this is just in the 80s, folks, the stuff I'm about to tell you was pipe dream stuff. Yeah, I can't look in your brain with fine spatial and temporal resolution down to millimeters and seconds and no. Without hurting your brain, no, it's not possible. It had never been possible in human history. So we had this huge revolution in things called PET scanning and fMRI. So naturally, we wanted to take a look at this question of amygdala, motion, memory with brain imaging. We started with this study in 1996, a PET scan study, in which we had people come into the lab, and they were scanned The scan is measuring how much their brain is sucking up glucose, how hard it's working. And just like your muscles, the more your muscles work, the more they suck up glucose. The more your brain regions work, the more they suck up glucose. So that's what the technique is measuring. So we had people measured while they were watching some boring films, some neutral films, or again, while they were watching some emotionally graphic stuff. And what we found was that while they're watching the boring stuff, the amygdala didn't seem to care. There was no relationship between how hard the amygdala was working while they were watching the boring stuff and how well they remembered it. But look what happened when we showed them emotional stuff. There's a beautiful relationship. Once things got emotional, the amygdala started to care. And the more the amygdala cared, the better they remembered 3 weeks later. So now what have I told you? I've told you adrenaline injection studies, arm and ice water studies, beta blocker studies, human brain lesion studies, human imaging studies, all converging on a single story. That when things get emotionally arousing, if there's an earthquake in this room right now, and things get emotionally arousing, and you remember this, and you remember the color of my tie, and you remember all kinds of stuff 15 years ago that you wouldn't, or 15 years from now, that you wouldn't remember, it's heavily, we think, because of the stress hormones fluxing like Holly Viola in the period afterwards, interacting with your brain, especially your amygdala, and telling other parts of your brain, remember this, it's important. Now here's where the trouble starts. I should have quit. Should have quit while I was ahead. Because about 10 years ago, I discovered that it's not the same in men and women. About 10 years ago, I was doing a PET scan study, and I noticed something funny going on. And it was that when I showed men and women the same emotional stuff and scanned them, it was the amygdala in the right hemisphere that was better at predicting whether the men would remember the stuff. And it was the amygdala in the left hemisphere that was better at predicting whether the women would remember the stuff. This is about 10 years ago. I'm an untenured assistant professor. I'm getting into something I know my colleagues basically don't think is very important. Sex differences, right? We'll get to the biases in a minute. But something in me said, I think there's something important going on here. And so I started to pursue it. It's 10 years on, now several people have found this. There is this intriguing sex by hemisphere difference in how the amygdala works in emotional memory. It's not the same in men and women, on average. Key word there, on average. All right? And it raises the question, what does this mean? What does that mean? And literally, I'll be working on it for the rest of my career. But it took me into the topic of sex differences in the brain, a topic that 10 years ago I couldn't have cared less about because I carried all the assumptions, implicit assumptions I was taught, implicit and explicit assumptions, which is that it's not a very important topic. But it turns out you can see how wrong I was. It turns out there's all kinds of sex differences in the brain. And as a matter of fact, when you work on this topic, people send you all kinds of funny joke slides. That's one of my favorites. And you hear people comment on this a lot. So, for example, here was a writer who was commenting on the difference between men and women. I love that one, too. Obviously, it's my talk, so I got to pick my favorites. But now here's a kind of an interesting one. Kate Whitehill, an Olympic soccer player, talking about the dynamics of soccer teams. She said, when a problem arises on the men's team, they punch somebody in the face and it's done. Girls, we hold it in. And when it comes out, it's fire, which is really awful. So you see comments like this all the time, and everybody's laughing because everybody experiences this and lives this. Here's the last of my favorite examples. It's brilliant. I call this my Arnold Schwarzenegger slide because when I was in graduate school, I was about 24 years old, and I had a female friend tell me something that was remarkable to me at age 24. She said, all women, when they look in the mirror, see what's wrong with them. I said, that's interesting. She said, what do all men see when they look in the mirror? And I said, Arnold Schwarzenegger. It's funny, but it also allows us to segue into the really important part. I gave this talk about 5 years ago on the East Coast at a place called Cold Spring Harbor, and in the audience was the director of the National Institute of Mental Health. And he came up to me afterwards and he said, can I have this slide? And I said, sure, why? And he said, because I have to give a presentation before a bunch of senators and congressmen on anorexia. And all of a sudden, it's not funny. All of a sudden, it's deadly serious. And that's true. The topic of sex differences in the brain is hugely important and hugely serious. Now, this takes me I'm into the second part of the talk now, sex differences in the brain. And I fear I'm going to run out of time because I'm having such a good time up here. But as long as you go away happy with having your money's worth, then I'm okay. But the second part now is, why is this topic so darn important? Well, it's because what the data are telling us, and I'm going to give you some flavor of that. But before I do, you have to understand that most of neuroscience was trained like I was, to believe that sex differences in the brain was pretty much what it was in 1966 when a famous guy named Seymour Levine wrote an article in Scientific American entitled Sex Differences in the Brain. Read the entire article and you will find one brain region mentioned. The hypothalamus, way at the base of your brain, really important in hormone control, especially sex hormone control. So for most of neuroscience, for most of my career, it's really changing now. Sex differences in the brain meant, a tiny little nucleus way at the base of your brain, some sex hormones and some sex behaviors. And the sex behaviors are different between the two sexes, so okay, something's got to be different in the brain. But it's not the neocortex, it's not the hippocampus, it's not the amygdala, it's nothing, important. And that was the view that certainly I was explicitly and implicitly raised on. And it turns out that view couldn't be more wrong. There are sex differences everywhere in the brain, at every level of function, at every lobe of the brain, in virtually all transmitter systems. There's sex differences in primate play behavior. There's evidence that female primates actually, when given a choice between playing with dolls and trucks, spend more time playing with dolls. And male primates spend more time playing with trucks. There's actual evidence of this. There's evidence of sex differences. You've all heard of knockout mice, probably. It's been a big thing for 15, 20 years. We're going to knock out a gene, see what happens. It's been the sexiest thing in biological medicine for 10, 15 years, knockout mice. Well, guess what? There's all kinds of examples. Lots of them end up in file drawers, but lots of them are published, where they knocked out the same gene in male and female mice, and they got something completely different. And they just go, because they have no idea what to do with that. So they publish the one that makes sense, and they put the other one in a file drawer. That's unfortunately not funny. But you get the point. And in fairness to them, people just don't know quite what to do with this issue. It's like exploding in the last decade. Oh, there's even sex differences in single neurons. The mechanisms of what's called apoptosis, the mechanisms by which neurons die in, for example, Alzheimer's disease, are not the same in male neurons and female neurons. They are similar and different. By the way, answer to the question, are men and women similar? Yes. Answer to the question, are men and women different? Yes. People have trouble kind of keeping both of those in mind. But the mechanisms by which neurons die are both similar and different in males and females. So you can see it extends, it's way more than the hypothalamus and sex behaviors. And let me just give you a couple examples from right here at Irvine. You've probably heard of Dr. Carl Kottman, one of our big Alzheimer's researchers. Well, 3 or 4 years ago, he was studying just how genes are expressed as people age. So he looked across a lot of ages of people, and he looked at gene expression, and he found that the patterns of gene expression were wildly different in different structures of the brain at different ages. It was a big gemisch, a big mosaic, depending on whether or not they were male or female. You might think that as people age, the same genes are expressed throughout your brain everywhere. And Carl found, no, that's not true. Here's another example from Irvine. Frances Leslie. She's studying what happens when you expose a fetus to nicotine. So, what does she do? She takes rats, pregnant rats, and she exposes them to nicotine. And she exposes the pups, in utero to nicotine. And she wants to know, what does that do to their brains, to their developing brains? And what you're looking at here, see the blue bars? That's once the pups grew up, how much were their brains expressing certain genes related to the white matter? You've all heard of the white matter. And you can see the blue bars? If they were exposed to nicotine in utero, when they grew up to be teenagers, their brains were expressing a lot more of these genes. Their brains were working differently, really regards to white matter. But wait a minute. That's only half true, because this is only the males. Want to see what happened to the females, the sisters? Oops. Now, what if Frances had only focused on the males? Her conclusion would be right and wrong. What if she had said, sex doesn't matter, tell her graduate student, put the males and females together, we build up our sample size, increase our N, and it doesn't matter. Now what would she have found? Nothing. She would have found that exposure to nicotine doesn't do anything to the expression of these genes. You see the point? And folks, this is one example. I can give you hundreds all throughout neuroscience. That's why I said it's a tidal wave that's coming at the field. Even our own Francisco Ayala, you probably heard him lecture right here, has gotten into this topic. He published a paper, a year or two ago, in which he looked at, he measured how brains were responding while people judged how beautiful certain images were. And he basically found that certain parts of the brain were bilaterally active in women, but only unilaterally active in men, making the same beauty judgments. So that's Francisco Ayala right here at UCI. And you may have heard, well, sure, there's sex differences in the brain, but they're small and unreliable. You can't really count on them. Is that true? No, it's not true. And if you were to ask another colleague here at Irvine, a very nice guy named Fritjof Krugel, who's extremely good at studying MRIs, ask him about what he calls the texture of the white matter of brains. How smooth is the texture of the white matter of the brains, and how ruffled is it? He found the difference so great that if you give him that number from one brain, he'll tell you 95 times out of 100 if that was a man or a woman. So the sex difference in the texture of white matter, forget what that means, we don't know, but I'm saying, are these sex differences all small and unreliable? No, they're not. They're big, middle, and small, and they're everywhere in the brain. Are they fundamental? I'll skip that to get on to the next part. But there are scientists who believe that by continuing in our animal models, in animals, I think 93% in neuroscience of the animals used are male. And there's people like this guy who studies pain, who's finding sex differences in pain perception, who's saying, given the sex differences we're finding in pain, one might argue that basic scientists are shirking their responsibilities to half the human population by avoiding direct animal models of them. We simply do not see any valid excuses remaining for the continued exclusion of female rodents from the basic sciences of studies of pain. Now, that was 5 years ago. I'm not sure what's changed. But the accumulation of data, this tidal wave, is gradually changing things. And honestly, it's a very exciting time, and it's a very good and important time. So I'll skip that one. Before I get into part 3 of my talk, I want to make one more point about this general phenomenon of sex differences in the brain, how ubiquitous it is, how it affects so much, and how we really have to wake up and start studying it better. And one of the reasons we really should is because it's actually what Charles Darwin would have predicted. And I didn't realize this myself till a couple of years ago. I've been a biologist for 30-some years, and I only actually started to read Charles Darwin about 2 years ago. It's almost a crime, because he's just such an intellect. He overpowers you. I promise you, go to Amazon.com, buy Origin of Species. Whatever criticisms you have, he already thought of them. He thought of them better than you. He thought of them more deeply than you, and he's answered them. He's a remarkable intellect. And anyway, I don't know why I'm selling you on Charles Darwin. Great guy. But I was taught that he said that evolution was driven by something called natural selection. Weren't you all taught that? It's not what Charles Darwin said. What Charles Darwin said, and he was crystal clear on this, was that natural selection and another force he considered a separate force called sexual selection drove evolution. He was quite clear that evolution without sexual selection fails. And what he was trying to explain was things like this. Charles Darwin knew darn well that according to his theory of evolution by natural selection, this shouldn't exist. This is a remarkably good way to get selected out. It is. Charles Darwin knew that. In fact, his grandson, these became popular in English gardens around the time of Charles Darwin, and his grandson said that every time his grandfather Charles saw one of these, it made him sick. Because he knew he couldn't explain it with natural selection, but he also knew that he could explain it with sexual selection. Natural selection is the force that acts on whether or not an animal survives. Sexual selection is the force that acts on whether or not an animal makes a baby. They're related. If you want to make a baby, it helps to be alive. But they are independent, and Charles Darwin knew that. And the reason I'm saying this, and the reason it's important for you, is that whereas natural selection, more or less equally acts on males and females, more or less. You can get sex differences from natural selection. Sexual selection, by definition, acts male on male or female on female. It's a force that, by definition, is going to produce sex differences in everything, in brains and in bodies. It's not going to magically stop at the neck, right? So that means if you believe in evolution as Charles Darwin actually described it, you should believe that it should produce brains and bodies of men and women that are full of similarities and differences. In other words, exactly what it's produced. Okay? So now we'll take us back into, that. Again, that's important because sometimes, my colleagues who were trained like me to think that evolution is all natural selection say, I'm having trouble believing this because I don't see why evolution should have produced these sex differences. And the answer is, actually it had to. It had to produce these sex differences that you're seeing out of results heavily of sexual selection pressures. Okay, so now let's take you into the third and final part of the talk. And it looks like I have this lovely clock up here so I can track myself. It looks like I've got a good 10 minutes left. Still trying to give you your money's worth. I want to take you back to the lab now. Emotion, memory, and the brain. And what have we found in my lab now about memory and sex differences in memory, building on this amygdala finding that started it all? Well, one of the first things I got interested in when I started to think about this was gist versus detail, forest, trees. I think in the interest of time and everything, I'm going to skip the setup, but just say that it turns out that the two hemispheres of your brain, it looks like they differentially process the gist and the details, the forest and the trees. And here's some evidence for that. See this little figure here? What is that? Made out of. Now look what happens if you take a person who has damage in his right hemisphere and ask him to draw that. Now look what happens when you take a person who has damage in his left hemisphere, or her, and ask him to draw it. Isn't that cool? Here's another one. What is this? Made of. Here's what happens when you ask a person who has damage in their right hemisphere to draw it. If you lose the right hemisphere function, you lose the gist. Here's the left hemisphere. Look at that. You lose the details. It's fascinating. And I decided to work with this, to run with this, and to basically marry it with my amygdala finding. And I'm going to skip the details for you, but just my hypothesis was, hey, maybe it's the case that the right amygdala in men, you know that sex difference I have in the amygdala? Maybe it's the case that the right amygdala is busy enhancing memory for the gist in men. Remember the wiring diagram I showed you with all those connections, the amygdala connected to everybody? What I didn't tell you is that almost all of those connections are from one amygdala to the same side of the brain, and from the other amygdala to its side of the brain. So each amygdala is basically going to be modulating what's going on in its own hemisphere. So I predicted that the right amygdala, maybe in men, is busy enhancing memory for more for gist of an emotional situation, but the left amygdala more is enhancing memory for details in women. I said maybe that's possible. And to test that, remember I told you at the beginning of the talk, the three-phase story with the beta blocker and the people didn't remember it, and I said there's a sex difference hiding in there? I went back to that data and I reanalyzed it with a prediction that the beta blocker, by blocking amygdala function, because you take it orally, but it's going to block amygdala function. It's going to do a lot of things, but it's going to block amygdala function. I said that beta blocker should have reduced memory for gist of the emotional story in men, but reduced memory for details of the same emotional story in women. And I went back to my published data and I reanalyzed it. And that's what I found. Blew me away. The beta blocker significantly reduced memory for the gist in men, but not women. Remember, phase 2 is where the emotional stuff happened? And the beta blocker significantly reduced memory for the details in women, but not men. This was the crossing the Rubicon moment for me. Because these data, probably when my career is over, the original data that I reanalyzed were probably the most well-known thing I've ever done. And hiding in them was this sex difference. And so at this moment, about 2003, I just said, okay, what are we doing here? We're missing something huge here. This was my crossing the Rubicon moment. So now I'm going to finish up by describing for you how we've been finding sex differences all over the place in emotional memory related to this in some ways, some of them, since, as I say, the blinders fell off. I'll skip that one. One thing we tried was we tried EEG. We said, how quickly do these sex differences start to arise when something emotional happens? And so we put electrodes on people's heads, and we recorded things that happen within 300 milliseconds. Things that happen in the brain within 300 milliseconds of seeing an emotional picture. And what we found was that men were showing bigger emotional responses in the right hemisphere than the women. And women were showing bigger responses, these electrical responses, in the left hemisphere. That's what this boring graph shows you. In men and women seeing the same slides, boom, record their electrical activity for 300 milliseconds, their head. Sex differences start to emerge. So sex differences start in how men and women are processing emotional things start within 300 milliseconds of something starting. Remember the business of dropping the arm in ice water? I hope you do. I should have dropped your arms in ice water. Well, a former student of mine, Joe Andriano, he wanted to know, is there a sex difference in that? So he had people listen to a story called The War of the Ghosts, drop their arm in ice water, wait a week, test their memory. Here's what he found. Did the dropping the arm in ice water in his experiment work? Remember, this is the first time we'd separated out men and women now. Here's the answer. Yes, it worked great. No, it didn't work at all. Yes, it worked great in the men. The men who dropped their arm in ice water ended up remembering more than the men who dropped their arm in... But look at the women. Nothing. Joe's next experiment was to say, well, might the sex hormones have something to do with this in women? Now, important point. Fluctuating sex hormones are absolutely, no doubt, a critical part of the explanation of sex differences in brain function. They are also, equally importantly, no way the whole part. And there's plenty of evidence for both of those. But for this next experiment, Joe wanted to say, are the sex hormones somehow a part of what's going on here? So he did the experiment again. This time, he did it only on women, and he did it in women where the hormone phases were defined. Because the first time we did it, we didn't pay attention to what phase of the menstrual cycle they were in. Low hormones, high hormones, low estrogen, high progesterone. And basically, to cut to the chase, he found that how well the stress hormone, remember the stress hormones enhanced memory? How well they enhanced memory depended on how the levels of the sex hormones. 3 different phases of the menstrual cycle, 3 different relationships between stress hormones and memory. And it was only in the high progesterone phase where there was a positive relationship between progesterone and memory. Now imagine that we hadn't paid attention to sex hormones, which we didn't in the first experiment. What would you get? You'd get a washout. Right? So this was quite exciting. Published this a couple of years ago. In all the studies of stress hormones, remember I showed you the rat study from 1975, the one that started it all? In all the studies of stress hormones and memory and where were you on September 11th or how does Holly Viola remember In all of those studies, there had never been a single study of whether or not the stress hormones did what they did depending on the sex hormones. And then we did the first, and that's what we found. Student of mine, Nicole Ferre, following up on this work, wanted to know, it's a well-established clinical fact that women tend to ruminate more. As a matter of fact, you saw that in one of the quotes. After negative, especially negative emotional events, women tend to ruminate more than men. And so my student Nicole showed women some negative films, rather unpleasant films. Then she asked, over the course of the next couple of days, how much did these films pop into their head? How much did they ruminate on them? And what she found was that they ruminated on them more at different phases of the menstrual cycle. Women who were in the high progesterone luteal phase ruminated more than either men or women in the follicular phase. Yet more evidence that the naturally occurring sex hormones are modulating memory formation for emotional events. Remember, almost everything I'm showing you, it's only been the last 5 years or so, and it's all brand new because nobody ever looked. I have way more questions than answers, but it's really fun. She found that the more the women had progesterone in their system as they watched the negative films, the more they ruminated on them. Nicole then did something, another student of mine, Nicole Irvine, found something very similar in another study. The more women had progesterone in their system, the better they were remembering some emotional material. Nicole Ferre then did something that Jim McGaw, my longtime colleague, described as heroic. If this is all true, that the sex hormones are busy modulating memory for emotional stuff, then maybe it's the case that a woman who has a highly emotional event, like a rape, a sexual assault, maybe the degree to which she forms an intrusive emotional memory will depend on the levels of sex hormones she happens to have at the time. This is a hard study to pull off, and Nicole did it by collaborating with Melinda Wheeler and their colleagues at a sexual assault crisis agency in Southern California. And what she did was, what Nicole did was she basically tracked women. Many of these women, of course, are given morning after pills. And these morning after pills are high doses of sex hormones, right? Designed to stop the pregnancy. But there's different pills that give different hormones. And what Nicole did was she took a lot of these women and compared how bad was their PTSD depending on which sex hormones they got, right? And you might think, you're not going to find anything, but what she actually found was that all the women who took some sort of contraception had fewer post-traumatic stress disorder symptoms 6 months later. And very exciting to us, it might not look exciting to you, but very exciting to us was how well the pill worked depended on which pill. And in fact, it looks like a pill that has progesterone in it, a pill that has combined estrogen and progesterone worked best. Now, actually putting that together with my lab results is hard. But one of the reasons I love this result, which is my most recent paper, was just published in the Journal of Forensic Nursing, is that this is a new piece of information that the nurses can use to start to decide which woman should get what pill in helping them to prevent post-traumatic stress disorder. So in case it's not clear, I'm telling you a story that goes from rats and mice running around mazes through college sophomores and brain imaging studies to women who are being traumatized and helping to figure out what it is that's determining how bad the memory consequences are. And one of the things appears to be the levels of sex hormones that are in the woman's brain in the period after the trauma. I'm very thrilled with this result. It may look boring to you, but I consider it one of the best things I've ever done in my career, because we've left the world of the college sophomore and we're testing this out in the real world. Another study to tell you about that was just published. A student of mine, Sean Nielsen, wanted to know, remember I told you that the sex hormones and the stress hormones are interacting to affect memory, the drop in your arm in ice water? Well, Sean wanted to know, okay, and during those studies, if a woman on UCI's campus was on the pill, on hormonal contraception, she was out. We didn't study her. Why? because I'm trying to understand natural sex hormone fluctuations and what, blah, 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 luteal, how they relate to. So they're out. But my student Sean said, what about the women on the pill? That's the basic question. If sex hormones are so important for memory, especially emotional memory, and if you wildly alter them, which you do with hormone contraception, shouldn't that do something to emotional memory? And I said, yeah, I'm not sure what, but let's look. So she showed women that same three-phase story, the emotional story, little boy gets hit, and she showed it to either some naturally cycling women or some women on hormonal contraception, all, of course, Irvine students. Then waited a week, tested their memory for the story. What she found was that in the naturally cycling women, they showed enhanced memory for the details of the story, but not the gist. Remember I told you the beta blocker blocked memory for the details in the women? So that fits perfectly. What about the women on the pill? They showed enhanced memory for the gist, but not the details. And she's recently finished a study of men, and basically the women on the pill are remembering it like men. The naturally cycling women are showing a consistent enhancement of memory for details, which is eliminated by lowering the sex hormones. Folks, this is brand new stuff. It has been 50 years since the first hormonal contraceptive was introduced by Searle Drug Company. 50 years. This study was published about 3 months ago. It's study number 1 of the effects of the pill on emotional memory. We're working on study number 2. So you can see, I hope I've made it clear to you, I've certainly tried to illustrate that this issue of sex influences on brain function, which I had thought was so boring, is huge. It's everywhere, and now I've got almost a cornucopia of findings on I don't know what to do with. But it's fun. It's also not just the humans, it's affecting the animal research. Jim McGaw's lab, several years ago already, here was this guy Cahill down the hall, and he's talking about the left amygdala doesn't do the same thing as the right amygdala. And one of his graduate students wanted to know, is that true in rats? So he trained up rats, and then he injected some drugs into their amygdala to affect memory. The same way that I inject adrenaline in the arm or drop your arm in ice water. He trained a rat up, or you can inject adrenaline into a rat. He trained the rats up, and then he injected some drugs right into their amygdala. Either their left amygdala or their right amygdala. And what he found is remarkable. So here's a drug called dopamine. It's a neurotransmitter. You've heard of it. When he injected it into the right hemisphere amygdala, this is a drug, this is a dose response and memory enhancement. And when he injected it into the left amygdala, nothing. Here's the next drug. This is a norepinephrine. The beta blocker acts at the norepinephrine system. Well, now we're going to inject a norepinephrine drug right into the amygdala. It boosts norepinephrine. When he did it into the right amygdala, that's a very pretty and memory-enhancing effect of the drug. And when he injected it into the left amygdala, this is rats. So this means that then this is all males, because nobody looked at females yet in the rats. Very few people do. But in the male rat, we're seeing an amygdala hemisphere deal, kind of like what we're seeing in humans. It's very impressive to me. It is true that all theory is gray. And as I've gone along as a basic scientist, I have wanted more and more to see clinical relevance, clinical impact. That's why one of the reasons I'm so happy with the work with the trauma that I told you about, and work related to PTSD, that kind of thing. But it's the relevance to the clinic that's really the final thing that makes this so important, because you're going to have trouble telling me of a disease that affects the nervous system that doesn't show sex differences, either in its incidence or in its nature or both. You're going to have trouble telling me a disease where I can't say, well, here's evidence for a sex difference. And for almost all of these, including, for example, autism, huge sex difference. For almost all of these, if you ask the relevant investigators, so why is there a sex difference? Here's the answer. I'm serious. That's the answer. And, maybe it was okay for a while, but it's not anymore. And that's one of the big stories I've got to tell you, is that sex differences in the brain are huge. They're omnipresent. They're little, medium, and huge. They're all over the place. They have the clinical implications are remarkable, and I'm really thrilled to be working on this topic now. I'm going to close with going back to my new favorite hero, Charles Darwin, with another wonderful quote from him. He said, "To kill an error is as good a service as, and sometimes even better than, the establishing of a new truth or fact." If all the scientists are operating under a profound error, it's really good to get rid of that error, even though you haven't established any big new fancy finding in nature. And that's what I like to think I'm doing. On the story of emotion, memory in the brain, and where was Holly Viola when that guy committed suicide, and where were you on September 11th, and where were you when your favorite baseball team won the World Series? I haven't experienced that yet, but where were you on all these highs and lows of your life? I think we're helping to remove an error. And the error is that you don't really need to care about whether it's a man or a woman, or whether we're talking about the left or the right hemisphere. And in so doing, I'm hoping we're going to advance the field. And I'll leave you with this thought. For 10 years, starting as an untenured assistant professor, I've been asked by the general public and by my scientific colleagues alike, why do you care? Why do you care about sex differences? And of course, what that question does is it puts the burden of proof on me, right? It says, hey, Cahill, justify this stuff. And I don't do that anymore. I have a little chip on my shoulder now. The data are so huge, I've given you a tiny fraction, so ubiquitous, it's so profound now, if you just open your eyes, that I politely say to my colleagues, the burden of proof has shifted. It is no longer the responsibility of me to explain why I'm interested in this. It is the responsibility of my colleagues to explain why they're not. And that is the position I'm sticking with. And I want to thank you very much for your attention. The people here are going to be coming around with buckets of ice water. I would like you to all